My dear students, we have already seen what is transcription, that is, formation or synthesis of RNA from the DNA template. Once transcription is over, I said the next process is translation, which is the synthesis of protein from mRNA template, right? But before this translation process, there is another intermediary process called post-transcriptional processing. Here, some modifications happen in the RNA before it acts as a template for protein synthesis. So, this is what we call post-transcriptional processing. Now, let us first understand this flowchart. The DNA is going to act as a template for the synthesis of RNA by means of a process called transcription. But in eukaryotes, initially when the RNA is formed, we are not going to call it as a mature RNA. We are going to call it as HNRNA. HN stands for heterogeneous nuclear RNA. And this RNA needs to undergo certain changes. And once those changes occurs by means of a process called post-transcriptional processing, we get the mature RNA. Here we are mainly talking about mRNA. So we get a mature mRNA. So we have two processes here. One is transcription, the other one is post-transcriptional modification. And when these two processes get over, we get the mature mRNA from the DNA. And these two processes occur inside the nucleus. And once the mature RNA is formed, the RNA will come out of the nucleus, will get transported to the cytoplasm, and there it will act as a template for translation or protein synthesis. So this is what happens in eukaryotic system. In prokaryotic system, we don't have such modifications happening. Directly, the RNA that is formed from the DNA will act as a template for protein synthesis. Whereas in eukaryotes, it undergoes another process called post-transcriptional processing before it acts as a template for protein synthesis. So that is what has been written here. In eukaryotes, the primary RNA transcript, as I mentioned, hnRNA, heterogeneous nuclear RNA, undergoes some structural and chemical changes after transcription to become a mature and functional RNA. This is then transported out of the nucleus to perform various functions. And these structural and chemical modifications that happens in mRNA are called post-transcriptional modifications. Students, we know that the mRNA is a linear RNA without any stem and loop structure, and it contains two ends. One is the 5' end and the other one is the 3' end, right? Now, what kind of changes that happens in mRNA during post-transcriptional processing? Three important changes occur. One is called 5' capping. This takes place in the 5' end of mRNA. The second modification is called 3' poly A tailing. This happens in the 3' end. And the last one is called splicing. Let us see the details of these modifications one by one. The first modification that we are going to see here is 5' capping of mRNA. Basically, it means the addition of a methyl guanosine to the 5' end of the mRNA. Let us consider this is the 5' end, and the 5' end will have triphosphate group here. To this 5' triphosphate group, addition of a methyl guanosine, that is guanosine is GTP. To this guanosine, a methyl group has been added at its 7th position. So this is 7-methyl guanosine. So this is a methyl guanosine which has been attached to the 5' end of the mRNA. So this is what basically means 5' capping. So an additional base has been attached here to the 5' end, and that base is a modified base, that is, it is a methylated base which has been attached. So this is what we call 5' capping. Now let us see a little more in detail about how this 5' capping happens. See, look at this diagram. Here we have the mRNA which contains the 5' end, and as I said, the 5' end contains triphosphate group in its end, and to this guanosine, a GTP will be added. Now GTP also contains 3 phosphates, so at its 5' end, the guanosine will also have a triphosphate group. Now this 5' end and this 5' end is going to interact. And during this interaction, the first thing that happens is the terminal phosphate of this mRNA 5' end. That is, in the mRNA, as I said, there are 3 phosphate groups. This is alpha phosphate, this is beta phosphate, and this is gamma phosphate. The gamma phosphate or the terminal phosphate of this mRNA will first be removed with the help of the enzyme RNA triphosphatase. Using this enzyme, the last phosphate will be removed and only biphosphate will be remaining here. This is beta phosphate and this is alpha phosphate, and the beta phosphate will now interact with the alpha phosphate of the guanosine GTP. ಸೊ GTP ಅಲ್ಲೂ ಕೂಡ ಆಲ್ಫಾ ಬೀಟಾ ಗಾಮಾ ಫಾಸ್ಫೇಟ್ ಇದೆ ಅಲ್ಲ, ಸೊ ಈ ಆಲ್ಫಾ ಫಾಸ್ಫೇಟ್ ಜೊತೆಗೆ ಈ ಬೀಟಾ ಫಾಸ್ಫೇಟ್ ಇಂಟ್ರಾಕ್ಟ್ ಮಾಡುತ್ತೆ. ವೈಲ್ ಡ್ಯೂರಿಂಗ್ ದಿಸ್ ಇಂಟ್ರಾಕ್ಷನ್ ಅಗೈನ್ ಅನದರ್ enzyme called guanylyl transferase will help in the formation of bond between this beta phosphate and this alpha phosphate. And during this process again the pyrophosphate from GTP will be released. So ಈ ಎರಡು ಫಾಸ್ಫೇಟ್ GTP ಇಂದ ಈ ಎರಡು ಫಾಸ್ಫೇಟ್ ರಿಲೀಸ್ ಆಗಿ ಬರೀ ಈ ಫಾಸ್ಫೇಟ್ ಮಾತ್ರ ಉಳಿಯುತ್ತೆ. So there is a bond formation taking place between the alpha phosphate of GTP and the beta phosphate of the mRNA. So again at the end of this what you will be getting is a structure like this. You have the mRNA which contains its alpha and beta phosphate which has been attached with the alpha phosphate of the GTP. So again you have a triphosphate formed here between the two 5' end, right? So GTP the 5' end, mRNA the 5' end illi interact madide. So it is a 5' triphosphate group formation taking place between these two molecules with the help of two enzymes. And I also said that this GTP is a modified GTP, that is, it is a methylated GTP where in its 7th position we have a methyl group, and this methylation is done with the help of the enzyme methyl transferase. So I have mentioned 3 important enzymes here. One is mRNA triphosphatase which removes the terminal phosphate of the mRNA. The second one is guanylyl transferase which helps in the bond formation between the beta phosphate of mRNA and the alpha phosphate of GTP. And the third one is the methyl transferase enzyme which adds a methyl group to the 7th position of the guanosine. So at the end of all this what we have is a capped transcript of mRNA which contains an extra guanosine which is methylated in nature. So whatever I said about 5' capping has been shown in written form here. 5' end of hnRNA is capped by methyl guanosine triphosphate. So what does that mean? There is an addition of 7 methyl guanosine in the 5' end. During this process what happens is one of the terminal phosphate groups is removed with the help of the enzyme RNA triphosphatase, leaving a biphosphate group in the 5' end of the mRNA, and then GTP is added to this terminal biphosphate with the help of the enzyme mRNA guanylyl transferase, during which a pyrophosphate will be lost from the GTP substrate, and this results in the formation of 5' triphosphate linkage. And at the same time the 7th nitrogen of guanine is methylated by mRNA methyl transferase, and for acting as a methyl donor we have S-adenosyl methionine. So what we have at the end of this 5' capping is an mRNA which has an additional methyl guanosine at its 5' end. Now just like the changes that happens in the 5' end, the 3' end also undergoes a major change which is called poly-A tailing. So in the mRNA, that is in the pre-mRNA, at its 3' end, large number of adenine residues are added. ಹೇಗೆ 5 ಪ್ರೈಮ್ ಎಂಡ್ ಅಲ್ಲಿ GTP ಆಡ್ ಆಯ್ತು, ಸೇಮ್ ವಿತ್ 3 ಪ್ರೈಮ್ ಎಂಡ್ ಅಲ್ಲಿ ATP ಆಡ್ ಆಗ್ತಾ ಹೋಗುತ್ತೆ. ಅಪ್ರಾಕ್ಸಿಮೇಟ್ಲಿ ಅರೌಂಡ್ 200 to 250 adenine residues are added one by one. So there is a continuous sequence of ATP that will be observed in the 3 prime end of the mRNA. So this is what we call poly A tail. And for this addition, we have an enzyme called poly A polymerase, which adds about 200 to 250 adenine units at the 3 prime end of using ATP as the precursor. The third important modification that happens in pre-mRNA is splicing. When the RNA is formed from DNA, and if you see the sequence of this RNA, it will have two important sequences in an alternative manner. One is called exon sequences, the other one is called intron sequences. Usually ಇದು eukaryotic RNA ನಲ್ಲಿ ಮಾತ್ರ ಇರುತ್ತೆ. Prokaryotic system ಅಲ್ಲಿ prokaryotic RNA ನಲ್ಲಿ ಈ ತರ ಇರಲ್ಲ. Fine. So we have exons and introns present alternatively in mRNA of eukaryotes. In this sequence, what we actually need is exon sequences, and the intron sequences should be removed. Why? The reason is exon sequences alle codons irodu, which will code for particular amino acids. Introns alli ee codons iralla. So ಈ exons ಮಧ್ಯೆ ಈ ತರ introns ಇದ್ರೆ ಇದರಿಂದ from this you will not get a functional protein, because ultimately RNA has to act as a template for protein synthesis, right? So the exon sequences should be present without any interference from the intron sequences. So ಈ intron sequences na remove ಮಾಡಬೇಕು, exon sequences na join ಮಾಡಬೇಕು for getting a functional protein out of this mRNA. And this process of removal of introns and joining of exons is called splicing. This splicing process is catalyzed by a very large protein complex called spliceosome. It involves both RNA and proteins, forming about 5 small ribonucleoproteins. So because it is made up of both RNA and proteins, we are going to call it as ribonucleoproteins. So 5 such ribonucleoproteins should come together to carry out the process of splicing. Now to understand this, let us see how the intron sequences are present, how the spliceosome recognizes the intron sequences. It mainly recognizes both the 5 prime end and the 3 prime end of the intron sequence. Usually introns alli 5 prime end alli ee tara GU anta ಒಂದು sequence ಬರುತ್ತೆ, ಒಂದು dinucleotide ಬರುತ್ತೆ. Same way in the 3 prime end it will have a dinucleotide, that is AG. And somewhere in between these two ends, it also has a A. Now these three important regions are recognized by the spliceosome protein, as whenever it comes across such a sequence, it will come and bind, especially in the 5 prime end, it will come and bind and it will cut the dinucleotide out of the exon. Exon ge GU ge madhyadalli ಏನು bonding ಇದೆಯೋ that will be first cut. And once the cut is formed, the GU is going to make a bond with the internal A that is present here. So ultimately what you will be having is a cut here, the GU forming a bond with A. You will get a structure like this. This is called a lariat structure, and this structure again will undergo changes where the spliceosome will make another cut in the AG region. And once that region has been cut, the whole intron would have come out and the two exons will be joined together. So ultimately what you see here is the spliced mRNA where the two exons have joined together and the intron has been cut and it will be degraded, further used for some other purposes. So to understand this whole process again in an animated version, you can go through this particular YouTube link, which I will send through notes. So in this, the splicing has been shown in a much simpler way to make you understand easily. So we have the pre-mRNA, which contains exons and introns. Uh, in the first reaction, what happens is the 5' end of the intron will be cut by the spliceosome, which will join with an intermediate adenine residue to form a lariat-like structure. And in the second reaction, this 3' end will be cut and the two exons will be joined together. So now this is the mature mRNA that is formed, which will undergo and which will act as a template for translation, whereas the intron will be discarded. So in the structural gene, which contains both exons and introns, the introns are removed and the exons are joined together to form the mature mRNA. Now let us understand the whole process of post-transcriptional processing with the help of this chart. So what at the end of transcription, what we have is the heterogeneous nuclear RNA, which will first undergo 5' capping, that is addition of 7-methyl guanosine at the 5' end of the mRNA, and then it will undergo 3' poly-A tailing, where the addition of 200 to 250 adenosine residues happen in the 3' end of the mRNA, and finally splicing, where the introns present in the mRNA will be removed and the exons will be joined together. So at the end of all these three processes, what we have is the mature mRNA, which will carry out translation. So again, the whole process explained in the diagrammatic way. We have the pre-spliced immature mRNA, which contains a 5' end and a 3' end and exons and introns. First addition of 5' capping and then polyadenylation of 3' end. So 5' cap is there, 3' poly-A tail is there, and removal of introns and ligation of exons happening. So all the introns removed and all the exons are joined together. So what we have at the end is the mature mRNA. Students, we have seen in brief how the post-transcriptional processing happens with eukaryotic mRNA. Now, a question might arise in your mind, why all these modifications should happen, especially 5' capping and poly-A tailing? Because splicing, we know that exon sequences only should be present to get a functional protein, and that is why intron sequences are removed. But first of all, why such intron sequences are present in the RNA? That has to be answered. That has to be questioned first of all. And also, why all this modification should happen? See, basically 5' capping and 3' poly-A tailing happens mainly to protect the mRNA. Why protection is needed for mRNA? The reason is, when the RNA is formed from the DNA, especially in eukaryotic system, this RNA is formed inside the nucleus, and inside the nucleus both DNA and RNA are protected. They don't have any threat. But as soon as the RNA comes out of the nucleus and goes to the cytoplasm for the process of translation, it will be attacked by nuclease enzymes. Because nucleases are present in the cytoplasm all the time, the RNA might get destroyed by these nucleases. And to protect these RNA from the nuclease enzyme, such modifications are very much essential. Both poly-A tailing and 5' capping is essential to protect the RNA from the nucleases present in the cytoplasm. Fine. Now, I will put other two questions to you to ponder over, to think. One is, whatever I told about the modifications that happens in RNA, it is mainly in the mRNA. You just find out whether such processing is necessary for tRNA and rRNA, and if it is necessary, what type of modifications happens in tRNA and rRNA. The second question is alternative splicing. What is this alternative splicing? See, when I say the exons are, the introns are removed and the exons are joined together, the process is not that simple and it does not always happens in the same way. Many a times this splicing happens in very different ways to get many different mRNA. So that process is called alternative splicing. Just go through these two processes and try to find out what are they. Thank you.